Okay, so in chapter 8, we're going to be talking about all things DNA. We're going to talk about the history behind the discovery of it. We're going to talk about the structure, how it's built, and then how it's built, how that relates to, and how the build of it relates to what it does. And it does two very important jobs. Number one, it has to replicate itself. And then number two, the information that it stores has to be turned into a protein or a product, because that is how DNA controls all the cell's activities, which is what we've been saying, since day one in biology. So, starting in section 1, we're going to look at some experiments that were done, some historical experiments that were accomplished and gave us some evidence that DNA is the genetic material. Now, these were not the only experiments, but these are the ones that were published and were the most sound of their time. So the first one we're going to talk about, now you got to remember that people did not know what gave people traits. I mean, science did not know how traits were passed from parent to offspring. There were lots of theories, but there wasn't any definitive proof of what it was. So Frederick Griffith, in the 20s, when pneumonia was a major problem, killing lots of people, he was studying pneumonia in his lab. And while he was studying pneumonia, he actually, and this is how a lot of things happen, scientists set out to study one thing but end up studying something else or determining something else. So he actually discovered what he called a transforming principle. So Griffith is not responsible for saying that DNA exists or anything like that, but he said that bacteria can be transformed by something. So he was studying, again, the bacteria that cause pneumonia, and he recognized that there were two forms of this bacteria. There was the S form, stands for smooth, because when you look at that bacteria under the microscope, it has a coat on the outside of the bacteria, and so it looks smooth. That form was actually the deadly or virulent or pathogenic strain that causes bacteria, or causes pneumonia. And then there was the second form called the R form. R stood for rough, because it did not have that coat on the outside of them, so they looked rough. And that form actually was not deadly, did not cause pneumonia, was not pathogenic. So what he did was a series of experiments to kind of see the effect of these bacteria on mice. He would inject the S form, and the mice, as you can see down here, right, inject the S form, and the mouse develops pneumonia and dies. He would inject the live R, which was not the deadly form, and of course the mouse lives. But then he decided, I'm going to heat kill. And when we heat kill things, that just simply means heat it up till it's dead. Just like when we have a boil order on, when there's a water main break in town, and you have to boil your water to kill stuff that might have gotten in it. That's what he did. So he heated up the S form to a point where all that bacteria will have died. And so when he heat killed the S bacteria and injected it into the mice lived. So he knew it had to be the living form of the S bacteria that, caused pneumonia. But then he did something else, and he took that heat-killed S bacteria that did not cause disease, and he combined it with the live R bacteria, which also did not cause disease. So he took these two things, these two bacterial strains that by themselves were not pathogenic, and he combined them. And when he did that, the mouse ended up developing pneumonia and dying. And then on top of that, he, when he performed his little mouse autopsies, he actually isolated out of that dead mouse live S bacteria, which is not what he put in. He put in dead S, live R, both non-pathogenic, mice dies, and they died from pneumonia caused by an S strain. So what he determined was that there was something being passed from that dead S strain to the live R strain, basically converting them into living S bacteria, making them deadly. So that was Griffith's experiment. So he said, hey, there's something that is transforming this bacteria. There is a transforming principle. But he never really identified it as anything in particular. So the next significant set of experiments were accomplished by Avery and his associates, McLeod and McCarty. And so what he did was he took Griffith's um, transforming principle, basically. He took that heat-killed S strain, live R strain, combined them, and what he did was he ran some tests on this material. And so he would take this solution that now caused disease, because it was heat-killed S, live R, and he would inject it into the mice. And of course, the mouse developed pneumonia and died. So then he would expose it to an enzyme. Maybe the enzyme got rid of proteins. And then he would inject it in the mouse, and the mouse still died. And then he maybe took out the lipids and injected in the mice, and the mice still died. So he's kind of doing this process of elimination that got him to the place where transformation kept happening no matter what he took out of that solution, whether it was carbohydrates or lipids or proteins or RNA. And the only time transformation stopped was when he broke down the DNA, when he used enzymes to break down DNA and got rid of the DNA. That's when the transformation stopped. So, and then he did a lot of chemical tests to analyze that, that material that he, isolated, and the chemical test proved that this material is DNA. So Avery basically said that DNA is the transforming principle. So, moving along, okay, we got this idea that it's DNA. Well, the problem is there were scientists who said, well, DNA doesn't have a lot of variety in it. It has like 4 parts to it: adenine, cytosine, guanine, thymine. There's not a lot of variety. Proteins, on the other hand, there's a lot of variety in proteins. So there was kind of a battle going on in the scientific community about, so what actually was the transforming principle? Was it the DNA or was it the protein, both of which are found in chromosomes? So Hershey and Chase, 2 more scientists, decided that they were going to study viruses that infect bacteria. We call those bacteriophages. So these viruses, clearly viruses are super small if they can infect bacteria. So these bacteriophages were used, and they were looking to see what part of the virus would get into the bacteria. Because basically viruses are a protein coat, a shell of protein with nucleic acid or DNA on the inside. And that's really all a virus is made out of. So they tagged or labeled, inserted radioactive phosphorus into the DNA of a culture of viruses. And the other culture of viruses, right, the other test tube of viruses, they labeled it with radioactive sulfur, and sulfur is in very high amounts in proteins. So basically, they had one culture of viruses, I like to think about it like they were glowing, glowing. One part of it was glowing, its DNA was glowing, and the other culture's protein coat was glowing. And so when they let those viruses then, this is the virus guy right here, when they let those viruses infect the bacteria and make more viruses, what they found inside the bacteria, right, which is what, we're looking to see what genetic material the viruses were giving to the bacteria. When they studied the bacteria, they found that it was DNA inside the bacteria, not proteins. So their experiment gave even more evidence to the idea that DNA is that transforming principle that Griffith originally discovered.